Meiosis, and what we remember about meiosis, the introductory part that we have written, meiosis ki hoy, it's a cell division, right? Mitosis is also a cell division. Second, it takes place in which type of cells or meiosis hoy? It occurs in germ cells or it occurs in what? It can be called it as germ cell or reproductive cell ba ji kwa no body cells or ki hoy mitosis hoy, right? Talking about the stages of cell division, whenever we talk about cell division, broadly divided into two stages, that is karyokinesis and cytokinesis. So both in both the cases, either it's mitosis or it's meiosis, duita process e hobo, karyokinesis hobo aru cytokinesis hobo. Talking about karyokinesis, again it has been divided into certain phases. Ki phases asile? Prophase, metaphase, anaphase and telophase. So these are the phases that would be observed both in case of mitosis and meiosis. Mitosis called as equational division, meiosis called as reduction division. Why is it called as reduction division? Because the number of chromosomes would become half as compared to the parent cell, the daughter cells will have half the number of chromosomes, right? So it is called as a reduction division. Next, talking about the process of meiosis, it gets completed into two phases or it is divided into two types, that is meiosis 1 and meiosis 2, right? What we will be studying is, so meiosis of hame ki boli koy, so reduction division, and you have to be precise, meiosis 1 is precisely the reduction division or meiosis 2 is the reduction division. Chromosome number kot komi jay, meiosis 1 not common and meiosis 2 common. Meiosis 2 is also called as equational division. So we understand this, that this is not a phase where the chromosomes will become reduced, right? So meiosis 1 ote hobo. And meiosis 2, I think you will remember we said that it is almost equivalent to ki mitosis, equational division hoy, or mitosis ko ami ki boli koisile, equational division. Okay, talking about meiosis 1 and meiosis 2, in between meiosis 1 and meiosis 2 there is a phase, that phase is called as interkinesis. Remember we have discussed a chart in the last class, right? So meiosis ki koisile je karyokinesis, cytokinesis, no? Kebar cytokinesis hoy aru kebar karyokinesis hoy. How many times karyokinesis takes place and how many times cytokinesis takes place in meiosis? Two times, right? Karyokinesis dubar hoy, cytokinesis dubar hoy. So meiosis 1 or meiosis 2 majot, there is a resting phase and that phase is called as interphase by interkinesis. Interkinesis. What is the similarity between interphase and interkinesis? Both of them are called as resting phase. What is the difference between the two? One point of difference, very important difference, DNA replication. No, DNA replication or DNA duplication occurs in interphase and not in interkinesis. No, in the process of meiosis, DNA duplication ke bar hoy? It's only one time, right? Saba okman introduction part of likhi disilo. Meiosis or DNA duplication ke bar hobo? Matra ebare hobo. No doubt we have two types of cytokinesis and two phases of karyokinesis. Kintu DNA duplication ke bar hobo? Ebare hobo, right? So that was the portion we have done, and we have started with prophase. Prophase or ki koisu, in case of meiosis, prophase is supposed to be the longest phase. It is further divided into five more phases. At our bit of the first phase, we have completed the discussion, and the first phase was ki leptotin, right? So whenever we talk about leptotin, what are the characteristics that you all remember? What happens to a cell during the process of leptotin? Yeah, very good. Since leptotin is a process of ki or is a phase of prophase, so prophase or like general characteristics, some of the characteristics you will see in this stage also, right? Fourth bench, ki hole, what happened? You, yeah, where are you? In the class? Okay, be in the class, okay? Okay, so leptotin or ki koisilo? Whenever we talk about leptotin, there are three words to be remembered. First, what is the meaning of leptotin? Leptos, no? Lepto or lemma ami discuss korisilu. So during this phase, the genetic material appear in the form of ki thin threads. So that's the first word you need to remember, right? Chromosomes dekha pua ne nupua? Can you see the chromosomes in leptotin? No, you cannot see the chromosomes. Chromosomes we don't see, but duplication of DNA হৈছিলে নে নাই হোৱা obviously হৈছিলে কাৰণ S phase of already হৈ আহিছে কিন্তু আমি chromosome দেখা নাপাওঁ chromatin network দেখা পাওঁ that's the first stage. Next, condensation of chromosomes takes place or it does not take place? It takes place. And if you talk about condensation, is it continuous condensation that takes place or it occurs at intervals? It occurs at intervals, and since it occurs in intervals, we have observed a structure called as A beaded structure, right? The genetic material will always appear in the form of beads. And what would the beads indicate? The beads would indicate that time or the area where condensation of the genetic material has taken place, right? Ekeloge hoi na jay, lahe lahe hoy, ba at one go hoi na jay, no? Intervals ot hoy, ekarone ami bead dekhabo. Next, the third word was something called as a bouquet stage, right? Question ki ahe, bouquet stage is said to be dash stage of prophase. Bouquet stage could do hoy leptotin. Why is it called as a bouquet stage? Because it looks like a flower, I mean bouquet of flowers. What we said is the genetic material, especially the chromatin, they remain attached with the nuclear membrane by their dash, by their telomere. And what exactly a telomere is? Telomere is the tip of a chromosome. And I think you all remember when we were discussing cell, we have talked about telomere, we have talked about chromosome. Telomere is something that's unique as compared to the rest of the chromosome, right? Guteidal chromosome tar tip to okoman hole unique hoy. Hey, uniqueness to karone ki hoy, the chromosomes do not get attached to each other. Tahate hodai separate hoy thake. So hekin loke koriso, we'll be continuing with the next step, that is aji kora zygote. For those students who have already read this chapter, there are certain words I think you all have already heard or you should learn about it. Words like synapses, words like bivalent, right? What is a bivalent? What is the process of synapses? What is synaptonymal complex? Question 18 ta word uporte ahe. Bivalent, synapses and synaptonymal complex. This is the portion that we are going to study in here. So up till now, jetia when we talk about prophase ot ki dekhisu, especially when we talk about leptotin, we have seen the structures, genetic material in the form of a thin threads and tat ki hoy asile, right? Next stage is zygote. What happens in zygote? Synapses takes place. What is synapses? Pairing of homologous chromosomes is called as synapses. So ebilak gute bilak ami ki boli kom chromosomes. So ebilak chromosomes ki hobo? Especially chromatin bilak condense hoy pele chromosomes form koribo, right? So chromosomes form korile eto structure hoy nohoy chromosomes or. It could be denoted as this, right? In the process of zygote, what happens is homologous chromosomes ki koribo? Pair koribo, right? Prothomete ki hoisile eta ke arrange hoy asile, tar pisot ki hobo? Pairing will start, and this process of pairing is called as ki synapses. And when we use the word homologous chromosomes, what we understand, homologous chromosomes mane ki? The chromosomes that are morphologically same, right? Eta chromosome ki hoy? Nothing but DNA. DNA yate ki thakibo? Genes thakibo. If you talk about this as a chromosome, yate ki thakibo? Genes thakibo. Homologous chromosome jedi ko, if we consider this pair as homologous, tetiyale ki hobo? Tar length to eke hobo, right? Tar jimanta number of genes ase hetu eke hobo. The position of the genes would also be the same. The position of the centromere would also be the same. Gute bilak jedi eke hoy, those pair are called as ki homologous chromosomes. So during the process of zygote ki hoy, jun bilak chromosomes scattered hoy asile, hey chromosomes bilake homologous pair bilake they will start pairing, and that process is called as synapses. Now if I say a pair is formed, right? Pair to form korile to nohobo no, tahate to log lagiu thakibo lagibo. Prothomete ki asile single asile, tapsot ki arambo hol? Pairing arambo hol. Now when I say that a pair has been formed, the pair needs to remain attached also, right? And this attachment is done by certain types of proteins, and that is what is called as synaptonymal complex, right? Synapses hobo, synapses or karone ki hobo? Homologous chromosomes or pairing hobo. Pairing hole ki hobo? Tahator majot kisman protein synthesize hobo that would form the synaptonymal complex. So the question that is asked is, in which stage of prophase do you observe the formation of synaptonymal complex? Zygote. Similarly, all of pisot the question would be synaptonymal complex dissolve ketia hobo. So accordingly, we will be studying. It needs to get dissolved. No? If you remember, meiosis at sobotse important event ki obviously crossing over, right? Once it has been crossed and the genetic material has been exchanged, the pair of homologous chromosomes will separate. And if it has to separate, then obviously this needs to get dissolved. So kuntu stage of form hoise, kuntu stage of dissolve hoise, what is the function, what is the process called as, these are the things that we are going to study. Together with that, we are going to study something called as a bivalent. Oh, likhai asu. Likhasu. Eitu loke to buji paisu no? Synapsisor karone juntu pair form kore, he pair to kami ki buli kau? It is said to be a bivalent. Right? So what is the important point about zygote? First, synapsis takes place. Second, formation of bivalent. Third, formation of synaptonymal complex. Now whenever we are talking about bivalent, by word to thakile, we understand we are talking about two, right? So bivalent related a question ahe. Eta cellot jiman chromosomes ase. If suppose if I talk about a human cell having 46 chromosomes, so in the stage of zygote, but zygote stage or by in the stage of prophase, tahate jodi bivalent form kore, keta bivalent form koribo. Yeah. So the number of bivalent is exactly the same as the haploid number of a cell. Eta cell, okay, human cell, n is equal to kiman? 46. Sorry, twice n is equal to 46. If twice n is equal to 46, n equals to kiman? 23. So 46 dal chromosome ase, tahate jodi pair form kore, keta pair kore, half. So that is another point that you have to remember. Question put a bivalent keta form kore. Bivalent keta form koribo, total jiman chromosomes ase tar half. Right? Or total jiman chromosomes as a half puli quadke. The better word that you will be using is, if twice n is that, n is equal to the bivalent. Yes. Right? One second. As you are writing the note, any point that you did not understand, please ask. Hey, can't I tell you, sir? Amount of DNA doubles, already became double. But you cannot see the chromosome. If the amount of DNA doubles, then we understand. I think you remember this diagram, no? Right? If amount of DNA doubles, then we should have observed this type of a structure. Kintu A structure to here dekha pam niki? Napam. Right? It was invisible starting from S phase itself. Right? এই দুটা লাইনটো বুজি পাইছো
Homologous chromosomes will have same telomere. Homologous chromosomes will have same telomere.
Please ask. Yeah, actually chromatid has already been converted itself into a chromosome, but since we do not see, right? So homologous chromosomes were to use kori aso, kintu it's now in the diagram what we see or under the microscope what we see, je chromatids bilak ekelage log lagi ase. When you are talking about a line structure or this thread like a structure, ami etu chromosomes boli nokoi pelai, instead ami ki call chromatid. Right? Why is it appearing like this? Does that mean DNA duplication did not take place? Hetu nohoy. DNA duplication already hoise, kintu clearly visible hua nai. So, ami ki koiso, no doubt we are using the word homologous chromosomes, kintu in the diagram, ami keneke dekhiso? Thread-like structures, homologous chromatid type. Ha. Dudal already ase. Kintu ami dekha poa nai. Aru condense hobo lagibo, titiahe clearly dekha pam. Obviously, saidal hobo. Right? Understood? Synaptonemal complex. SYNAPTONEMAL. Synaptonemal complex. Okay? So তেনেকে leptotinot we have to remember 3 words ঠিক তেনেকে some important points about zygote. What you need to remember about zygote is the word synapses, synaptonymal complex and bivalent, right? Question ahe, synapses occurs in which phase or it is seen in which phase? Formation of a bivalent is a characteristic of which phase of meiosis? So hey word gita monot rakhibo lagibo and respectively the stage where you can find it, right? So any point that you did not understand up till now? Clear? We are starting a cell that is ready to divide, and which process of division? Meiosis. At the starting itself, the genetic material appear in the form of a thin thread, and that stage was called as leptotin. After that, with slightly condensation started place, right? And then in the next stage what we have seen is homologous chromosomes started getting paired, forming a bivalent by the process called as synapses, by the formation of a particular complex that is made up of proteins and RNA, and that is what is called as synaptonymal complex, right?
In the process of forming bivalent is synapses.
The process through which a bivalent is formed is called as synapses, right? Is it over? Okay. One important point. Zygote is a part of what? Prophase. What happens in prophase? General points when we talk about prophase, the first point that we remember is the nuclear membrane disappears, right? But nuclear membrane, nucleolus, centrioles remains as it is. Mane even at this stage you can see a proper nuclear membrane. Nuclear membrane etialoke disappear kora nai. Ketia koribo? Last phase of prophase, right? Because this is just the starting. Leptotin is the starting. This is just the second point. At the second stage itself, the nuclear membrane does not disappear. Similarly, the nucleolus also does not disappear. If you are talking about centrioles remaining as it is, what exactly are we talking about? Nuclear membrane remains as it is. We understood it does not break. Nucleolus remains as it is. We understood it did not break and it did not disappear. If I say centrioles remain as it is, what did you understand by that? Yeah, actually they don't start moving to the two opposite poles. And the spindle fibers to already form hoye goise no, kintu tahate jot asile tate ase. If you talk about a centriole, where exactly can you find the centriole? Just nearby the nucleus, right? It is during the process of cell division that the centrioles will move towards the two opposite poles, right? Etialoke move kora nai, kot ase tar usorte ase, right? Ha আমি কি পঢ়ি আছো ৰিপ্ৰডাক্টিভ সেলছত, জাৰ্ম সেলছত, ৰাইট? তাত কি ক্যাৰেক্টাৰিষ্টিক্স থাকিব? এটা পেৰেণ্ট, প্ৰত্যেকটো সেলতে এটা পেৰেণ্ট, এটা ফাদাৰ, এটা মাদাৰ, মেটাৰনেল ক্যাৰেক্টাৰ থাকিব নাথাকে? সো সেই বস্তুটো পেয়াৰিং ক'ত হ'ব? অনলি ইন দ্য প্ৰচেছ অফ মিয়োছিছ। মাইটোছিছতো ফাদাৰ মাদাৰ দুইটা ক্যাৰেক্টাৰিষ্টিক্স থাকিব, কিন্তু তাহাঁতৰ পেয়াৰিং নহয়। এটা সেলত ক্ৰোমোজোমছবিলাক যে আছে, সেই গোটেই এটা তোমাৰ বডি যিমানবোৰ ক্ৰোমোজোমছ আছে, ইট বিলংছ টু হুম? ইট বিলংছ টু ইয়ৰ পেৰেণ্টছ। এজনৰটো নহয়। So প্ৰত্যেক দুইজনৰে দুটা দুটা এটা হেৰি আছে, কিন্তু this pairing process or synapses is only seen in case of meiosis and not in mitosis. A pairing process to অকল meiosis-তে হয়, mitosis-ত নহয়। Mitosis-ত the fact that we are talking about is about each and every individual chromosome. ইয়াতে কি? A pair of chromosomes. Mitosis-তো হয়, prophase, anaphase, telophase সব হ'ব। গোটেইবিলাক ক'ত হ'ব? এটা ক্ৰোমোজোমত হ'ব, ইণ্ডিভিজুৱেল। ইয়াতে কি হ'ব? দুটা দুটাই এটা পেয়াৰ কৰিব, তাহাঁতে প্ৰফেজ আণ্ডাৰগো কৰিব, তাহাঁতে এনাফেজ আণ্ডাৰগো কৰিব। both together or single. Single in case of mitosis, both together in case of meiosis. Right? That does not mean যে অকল meiosis হোৱা চেলটোতহে ফাদাৰ আৰু মাদাৰৰ characteristics থাকে। Mitosis হোৱা চেলটোতো ফাদাৰ আৰু মাদাৰৰ characteristics থাকে, but synapses does not take place. It is unique to meiosis. 23 pairs? এটা সেলৰ পৰা হৈছে। So ultimately আমাৰ লাইফটো যোনটো সেলৰ পৰা ষ্টাৰ্ট কৰিছে, that's a process of reproduction itself. So meiosis আণ্ডাৰগো কৰিছে। So meiosis আণ্ডাৰগো কৰিলে তাত পেয়াৰিঙে হৈছিলে, ৰাইট? পেয়াৰিং হৈ পেলাই ছেগ্ৰিগেট কৰিছে, ৰাইট? অ'কে, এইটো পইণ্ট লিখিলা? এইটোৰ ওপৰত কুৱেশ্যন আহে, পিছত আমি কুৱেশ্যনটো কৰিম। Next, pectin or pectin, G-qua. And what is so very important about pectin, as far as the different stages of prophase is concerned, the most dramatic phase boli kole, it is always the pectin that is considered. Why? Because the process of crossing over takes place in pectin. Crossing over kiho karne lage? Is it important or it's not important? It's very important. Why is it important? Because that is the basis of evolution. যেতিয়ালৈকে sexual reproduction নহয়, যদি sexual reproduction নহয় তেতিয়াহ'লে exchange নহয়, যদি exchange নহয় evolve নহয়, right? So obviously pectinot কি হয়, the most important process that is called as crossing over. If you talk about crossing over, what we understand, that দুটা homologous chromosome, they will exchange their genetic materials or their parts, right? So up till zygote we have seen something like this. ইয়াৰ পিছত কি দেখা পাম? So after this, we'll write it later, draw the diagrams later, okay? Just understand first and then we'll go for the note. Etu hol. Next is, ebilak chromosomes বা genetic material ki hobo? Since we are talking about the entire process of prophase, gutei process to ki hobo? Condensation to hoboi, right? So this time, is the point understood? Right? Actually, enugua to yate form already hoye goisile, but we could not see it. This time, this will become visible, right? So this is what is called as a dyad, D-Y-A-D, and two dyad would form a tetrad. Because এইবিলাক আমি পঢ়িম. And whenever we are talking about this pair, no, এই chromosomes to কি থাকিব? If you are talking about a pair, we have something called as sister chromatids. Sister chromatids, non-sister chromatids, right? When we talk about crossing over, jihete pectinot sobotse important ki crossing over hobo, crossing over kar mazot hobo, ei duta pair tur mazote hobo, right? So we have to use the word sister and non-sister. What we need to remember is, if you are talking about crossing over kar mazot hoy, sister chromatids and non-sister. Non-sister, if you talk about non-sister, what we understand, what is non-sister? Eta centromere jodi lagi ase tetehoলে sister, beleg centromere, that is non-sister. So if you are talking about this one and this one, tahatok ami ki boli kom? Non-sister. But if you are talking about this two, sister, ei duta ki boli kom? Sister. Yar pisot ki hobo? If you are talking about this one, don't draw, okay? We'll draw it later. Something like this, they will overlap, right? Overlap kori pelai, about breaking, shaking hobo, a part of them, it will get attached and etc. etc. Ultimately, ami ki dekha pam? Don't draw it now. If the process is understood, then notes we can write it down. Understood? Did not understand? Yes. Understood? Huh? So yate ki asil? Full red, sorry, full red asile, right? Etu ki asile? Full black. Yar pisot ki hol? Black or logot red lagi gol, red or logot black lagi gol. What does that indicate? Exchange of genetic material. What next? Separation. After the exchange has been done, obviously chromosomes bilak to eneke na thake, they needs to separate. So that is what happens during the next phase. Pectin or pisot next phase ase, no? So it is called as diplotin, and if you heard the word called as chiasma formation or chiasmata formation, the X-shaped structure, right? So what happens in pectin? This is something that happens in pectin. Clear? Let's write the note. Whatever we have explained is written in the form of a note now. Okay, have you heard something called as recombination nodules? So, pectininot aru eta word important, the appearance or the formation of the recombination nodules. Recombination, by the word itself, what we are talking about is nothing but crossing over. Recombination nodules ki hoy, it is the site where the crossing over will take place, right? Precisely tetrad, কিন্তু আমি tetrad use নকৰি পেলাই homologous chromosome as a whole কৈ থৈ দিওঁ, between a tetrad হৈ যাব. So what is crossing over? It is nothing but an exchange of genetic material process, and obviously certain types of enzymes are involved. We have talked about recombination nodules. So eta chromosome by homologous chromosome to jetialake recombination nodules does not develop, crossing over will not take place. So before the process of crossing over, it's very essential that recombination nodules needs to develop. So recombination nodules kenike develop koriba, some enzymes are associated, and what is the precise enzyme that we have to talk about is recombinase. So দুটা chromosome log lagi pale exchange koribo, exchange kora mane ki some parts of the chromosome will break down and then it will get attached with the other one. So breaking of the chromosome, attaching itself with the new pair of chromosomes, ei gutei bilak process ki hoy, enzyme mediated. You have certain types of breaking enzymes that will help in breaking. We have a word called as nicking. Ebilak bostu genetics or basic clear ke poriba, right? As of now, what we understand is if crossing over has to take place, there should be certain roles of particular enzymes. Enzymes that are responsible for the formation of the recombination nodules, enzymes responsible for the breaking of the chromosome, enzymes responsible for attachment of the new part. Okay? Ekini hol, ekini to ami bujisu. Ekini? Okay. Over, eto hoy gol? Yeah, the same process. Crossing over of karone ki produce kore, recombinants produce kore. Recombinants produce kora process to kami ki boli ko recombination. Yate jeti jega ase nohoy, yate aru eta enzyme likhibo lagisile. If you have space, if you don't have space, you can write it later also. Space to nai sake? Ase? Jar nai ki belek ke likhiba, ha? A point to hole niki? Last point? Nai howa no? It will keep us on the enzyme responsible for the development of breaks in a chromosome is endonuclease, right? Ami ki porthoise recombinase porhisu, and now we have endonuclease. Endonuclease ki koribo chromosome to bhangi bole chesta koribo, and this process of breaking a chromosome is called as nicking. Kot likhim? A2. Recombination in the homologous chromosomes is completed by the end of the pectining, leaving the chromosome linked at the site of crossing over. Crossing over is the formation of new characters. Okay, first come to this one. Crossing over means exchange of genetic material between whom? Between two chromosomes or between two homologous chromosomes. If materials are exchanged, then what will be the outcome? New characters would develop, but new form of genes, but new form of chromosomes would be formed. That is what is called as a recombinant, right? So recombinant jiman hobo, himan variation hobo nohoy. Variation hobo, our variation jiman hobo, himan ki hobo. Evolution hobo, so evolution hobo lagile important key. Crossing over hobo lagile important key. Sexual reproduction. So sexual reproduction important here. Karon it is the basis of evolution. Kiho karone sexual reproduction of key produce hoy? Recombinants produce. Understood? A2 understood now? Okay, come to this one. Okay, talking about this point, no? What we are talking about is that two chromosomes will exchange the genetic material. What will happen next? Duta chromosomes asil genetic material exchange korile. Will they remain like that forever, or they will tend to separate? Obviously, they will tend to separate. So after the exchange is over, ki hobo? Exchange over ketia hobo? Pectininot. After that, ki hobo? At that instant itself, will they separate, or they will just gradually try to separate? Gradually try to separate. So at that time, once the crossing over is over, what we will see, the two chromosomes being attached at the site of crossing over, right? Ei duta jodi chromosomes hoy, jodi crossing over hoise, duita junkhini jegat crossing over hoise tat lagi thakibo nalage. So this is the meaning of this line. They will remain linked up at the site of crossing over. Right? So crossing over hoi joa loge immediately pura eneke separate hoi najay. Prothomote log lagi thakibo, lahe separate koribo, lahe separate kora process tok, stage tok ami pectin noko. It is the next stage, diplotin. Right? The process of breaking of a chromosome is called as breaking of a chromosome mane swa eta chromosome bhangilehe tat beleg etukur part log lagibo paribo or no exchange of genetic material mane ki one part of one chromosome get attached to the other part of the other chromosome, yes or no, right? So eta chromosome jedi belegor part eta log lagibo lage tetia hale original chromosome dal okoman jegat break eta develop koribo lagibo na lage. That process is called as nicking, or what we say is chromosome showing nick, but that area where the breaking you will see, and the process is called as nicking. Break hobo, tar pisot exchange hobo. Break hobo, tar pisot attach hobo. Tak ami ki buli ko? Nicking buli ko. Understood? Okay, chromosome nicks would develop. Ketia develop koribo? Crossing over a homo develop koribo. Nohole tu, there is no point of breaking of a chromosome. Why should a chromosome break? Right? It is breaking because it has to undergo the exchange, and the process is called as nicking. Right? Just a rough diagram. If you want to draw, you draw. If you don't want to draw, don't draw, because understood, na? Diagram, you can see it from your NCERT book also. So yate ami crossing over to mane ki exchange to dekhwa nai. We have just showed that the two homologous or non-sister chromatids will overlap, no? Overlap kori attach hoise. Crossing over by exchange of parts to etialeke dekha nai. The diagram in diplotin would talk about the exchange. Huh? Oh, duta hoise, ekinto hoise, duta jekat hoise. Okay, starting from the beginning, leptotin, thin threads. Second point is beaded structure. Third point, bouquet. Zygotin, synapses, synaptonymal complex, bivalent. Pectitin, dyad, tetrad, crossing over. Clear? And recombinant, obviously. ইয়াতে নিউক্লিয়াৰ মেমব্ৰেনখন আকাই নাই দেখোন Okay, talking about diplotin. So, ekhin loke to buji palu, crossing over hoi jabo, and what will happen is the next diagram, no? Half of black, half of red, half of red, half of black. Tenuka hote jabo. After the exchange has taken place, they will try to separate. And whenever we are talking about separation, so duta chromosomes ase, if you are talking about this as a pair of homologous chromosome, tahate ki koile? They got attached at the recombination nodules, they exchanged their parts, and now they are ready to separate. Whenever we are talking about separation, it is not like this that they will separate instantly. এনেকে সেপাৰেট নকৰে ঠিক আছে দুটা ক্ৰোমোজোম আছে এই দুটা ক্ৰোমোজোম এনেকেও সেপাৰেট কৰিব পাৰে অৰ ইট কুড সেপাৰেট ইন দ্য ফৰ্ম অফ ওপেনিং অফ আ জিপ বেগ এটা বা কোবাৰ চেইন এটা যেতে ওপেন কৰা কি হ'ব লাহে লাহে ওপেন কৰে নকৰে ইট ইজ নট লাইক দিস দ্যাট দে উইল সেপাৰেট হোৱাট দে উইল ডু দে উইল সেপাৰেট গ্ৰাজুৱেলি মেবি দে উইল স্টাৰ্ট ইট ফ্ৰম দিস পইণ্ট নো অকমান বেকা হ'ব ultimately what they will form is a cross and this cross is called as ki chiasmata. So, the point is chiasmata formation is seen in which stage of prophase? It is seen in diplotin. Second, chiasmata ketia form kore or it is the result of what? It is the result of separation of the chromosomes after crossing over has taken place. Right? And whenever we talk about separation, it is not like this. It is the gradual separation and if it opens gradually, then at a point they will meet. That structure would be a X-shaped structure and that is what is called as chiasmata. Right? Hekhini loke ketia hobo diplotin. After diplotin, yeah, please ask. Etia to already akise hekarne moi aka nasilo. Right? Etia eneka nohoy tahate mane dekhabo to lagibo no je duita log lagise. So I have drawn this. Right? Proper diagram you cannot be talking about it. This X you will see only in the diplotin stage and plus this is not the diagram of diplotin. Why? Because the exchange I have not shown. Right? Jodi exchange dekhai pele eneke dekhao no etu ei jega kinik ami ki boli kom X shape that is called as chiasmata. Right? After diplotin Diplotinot কি হয়? The separation starts. Ultimately, they form a X. The next stage, that is dikinesis, no? Diplotinor pisot ki thakibo. Aru eta stage thakibo, that is called as dikinesis. Dikinesis ki hoy? When we talk about that chiasmata code lagi asile in the center, right? Gradually, it will move to the terminal position. Terminalization takes place in dikinesis. This is the only line that you remember about dikinesis. What is terminalization? Terminal point or duita chromosomes lagi thakibo. That is what is called as terminalization, right? They will tend to separate. While separating, at a point lagibo, asa ako kori pele eneke at a point they will remain. This is called as attaching itself in the terminal position, and after that they will separate. So a terminal position kot hobo, dikinesis hobo, X shape structure kot hobo, diplotinot hobo. Clear? Chiasmata is what is chiasmata? It is a X-shaped structure that is seen in the chromosome. We know there are two chromosomes of a homologous pair. They have exchanged their parts. A ronga to boga hogol, boga to ronga hogol, kibata to hobo no exchange. Exchange kora pisor what happens? They will tend to separate. Now talking about separation, either they can, so once the work is over, they can separate it like this also, right? Or they can gradually separate. Right? What happens to this pair? They will gradually separate, and this separation could be considered or can be compared with opening of a zip. Bag at a zip, it's not the entire thing that opens instantly, right? So, yate ki hobo lahe it will start opening, start from this part, tolor pra uporle jabo. So, it will start at a moment. X hobo. This X, a structure to jete form koribo, we say that this is what is called as chiasmata, right? And gradually, this X reach to the terminal position, terminalization. Oh, diplotinor পৰা start হ'ব. Pectitinot separation নহয়. Diplotinor পৰা gradual separation আৰম্ভ হয়. একদম শেষ কেতিয়া হ'ব? Last stage, that is dikinesis. Clear? Terminal positionত লাগি থাকিব মানে ইনফিনিট টাইমৰ কাৰণে লাগি নাথাকে ন? প্ৰথমতেতো এইখিনি পজিশ্যনতো লাগি আছিলে, হয় নহয়? কিন্তু লাহে লাহে খুলিলে নাই, তো টাৰ্মিনেল পজিশ্যনতো লাগি থাকিব, কিন্তু খুলি যাব। ক্লিয়াৰ? And if it has to separate, তেতিয়াহ'লে কি হ'ব? Synaptomimal complex dissolve হ'ব লাগিব। Form কেতিয়া হৈছিলে? যেতিয়া তাহাঁতে জইন কৰিছিলে, that is what is called as a bivalent. Where did it occur? It occurred in zygote. That protein complex needs to get dissolved, and only then they will start separating. So separation code হৈছে diplotin, right? So dissolution of synaptomimal complex code হ'ব diplotin. যেতিয়ালৈকে dissolve নহয়, হা? বুজা নাই? দুটা ক্ৰোমোজোম একেলগে লগ লাগি পেলাই পেয়াৰ হৈছিলে মানে তাহাঁতে এনেকৈ আছিলে, ষ্টাক টুগেডাৰ আছিলে, ৰাইট? কোনে ষ্টাক কৰিছিলে? সাইনেপ্টোনিমেল কমপ্লেক্স। যেতিয়ালৈকে সাইনেপ্টোনিমেল কমপ্লেক্সটো দুইটাৰে মাজত আছে, তাহাঁতে ছেপাৰেট কৰিব পাৰিব নেকি? নোৱাৰে। তো সেই ব্ৰিজটো ইউ নিড টু ডিজলভ ইট না। চ' ৱেৰ ডাজ ইট টেক প্লেচ? ইট টেক্স প্লেচ ইন ডিপ্লটিন। ক্লিয়াৰ? পেকাইটিনতো থাকিব, কাৰণ ক্ৰছিং অভাৰ এটা পাৰ্টিকুলাৰ পইণ্টতহে হ'ব। গোটেই ক্ৰোমোজোমত নহয় নহয় গোটেই ক্ৰোমোজোমত দুইটা লিংক হৈ থাকিব যোনটো পইণ্টত ক্ৰছিং অভাৰ হ'ব সেইটো হ'ব ৰাইট হা য়েছ At a point called chiasmata, Chaismata, chiasmata or chiasma, ji kwa. Did you get the spelling? Okay. এখানে এক্সট্রা পয়েন্ট দে oocyte नहीं बोलेंगे I was about to ask that question itself because this is something that we have already done in cell. Remember we have talked about special type of chromosomes. Remember lamb brush, right? So oocyte, the egg or that cell that will form the ovum, ultimately will form the egg, That is what is called as oocyte. DICTYOTENE. Ekini extra day, not the characteristics of diplotin. It is just to say that in certain organisms, diplotin might stay for months or years. Diplotin staying for months and years. What we understand by that statement? If I say diplotin has lasted for months or it has lasted for years, tar mane ki? Separation nahi hua, right? Jodi separation nohoy, remember we have talked about it, Lamb-Brush chromosome and salivary gland chromosome. Separation nohoy, DNA content bahi goi thake, tetihale ki hobo? Abnormal content bahi jabo. So certain case of ki hoy, we have seen Lamb-Brush. Lamb-Brush is a result of ki extended diplotin, right? That is seen in certain organisms and precisely in which cell? Oocyte, right? So while tending to separate, the point at which they remain attached is called as chiasma, right? When they are separating, at a point of lagi thakibo X shaped and tak ami ki boli chiasma. This is another chiasma. Chiasma ketia form koribo jete separate koribo arambho koribo, right? Jetia lake separate nokore tele chiasma form nokore. Oh yes. Just a second. Leptotin, zygotin, pectitin, in all the three stages, the nuclear membrane will remain intact. Partial degradation corpora hoise, diplotin onwards, right? And so in dikinesis, you will not see the membrane at all. Oh, sorry. Degradation. Degradation. এটা হল না So this is the last phase of prophase, and since we are talking about the last phase, so Professor GZ Hoy, all the things that you will see: dehydration, yes; spiralization, yes; next nuclear membrane, nucleolus, all the organelles disappear; condensation of the chromosomes properly visible in case of prophase, right? Once prophase is done, if the nuclear membrane disappears, then recombinants bilak ki hobo, it will get scattered in the cytoplasm, and then it will move to the next phase, that is metaphase 1. At that time they will be aligned in the equatorial plan, right? So in the next class we will be talking about metaphase. As far as this point is concerned, a point to monot rakhiba. So no doubt we have written a very lengthy note, but if you see, it is only a few points against each stage that we have marked, right? So hey stage kita extra ke likhi loba ba extra ke monot rakhibole chesta koribo. No point, for example in dikinesis iman khini poribo dokar nai to. These are the points that talks about prophase itself in general: spindle fiber, astral rays, ester, etc., etc. So dikinesis a gutte hupar bhitor what you need to read is that just one word: terminalization, right? So mark kora kita poriba. I think that's all for today. We'll meet you in the next class. Yeah, please ask. Astral rays, astral rays তুমি আগৰ ক্লাছ কৰিছা নাই? কৰা নাই, কিয় কৰা নাই? এটাও কৰা নাই, চেলৰ ক্লাছেই কৰা নাই কিয়? You are not well or something like that. Astral rays ক্লাছ মিছ কৰিলে প্ৰব্লেম হ'ব দেই। সেন্ট্রিওল বুঝি পাও? সেন্ট্রিওল কি হয়? Remove your mask, I cannot hear. Okay, it is the site from where the spindle fibers originate, right? So centrioles are pura, some fibers originate, and if you talk about the fibers this one short or kisuman long ase, right? If the fibers are short and plus appear like a star, that is what is called as astral, right? The entire structure is called as the ester, and these fibers are called as astral rays. Corpora ulai, centriolopora ulai, ketia ulai during the process of cell division ulai, right? And as far as these rays are concerned, either it is spindle fibers or astral rays, it is made up of what? Microtubules. Microtubules we have already completed in the class. Okay, that's all for today. Thank you. Thank you, thank you, thank you, thank you, thank you. Same to you. I think Monday we have a class. No, Monday class is no. Advanced. Aji, enjoy. 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 Very much essential.